Hey everybody, my name is Alexandra Van Dorsten and I will be doing a presentation on rabies. So just a quick introduction. The rabies virus belongs to the lysovirus genera, which includes several viruses that infect animals. These include the Lagos bat virus, the Macola virus, the Duvenhage virus, and the European and Australian bat viruses. The rabies virus is a negative sense single-stranded RNA virus, which just means that its genome is in a form that is complementary to mRNA. mRNA is considered positive sense, so the complementary strand is negative sense and must be translated to positive sense before transcription occurs. The rabies virus is transmitted from animals to humans, usually through dog bites, but other rabid animals may also transmit the disease. So it's possible to contract the virus through a bat bite or a raccoon bite or a fox bite, anything that the virus exists in. Rabies is a relatively rare disease in humans, especially in developed countries. The United States, for example, only reports 1 to 3 cases per year. This number greatly increases in less developed countries, but still affects fewer people than other communicable diseases that can be transmitted from human to human. Rabies is 100% preventable by avoiding rabid animals and also by getting a rabies vaccination. But as soon as the symptoms begin to show, there's very little chance of survival. So all of the treatment for rabies is preventative. The rabies virus affects the central nervous system and primarily replicates within neurons. Rabies is a zoonotic disease, meaning that it is transmitted from animals to humans. It can be passed to humans through any kind of open wound or mucous membrane that comes in contact with contaminated saliva. 99% of the time, this is a dog bite, but it is also possible to contract the disease if contaminated saliva enters someone's eyes, nose, or mouth, which are all mucous membranes. This is very rare, but it may be an occupational hazard for those working with animals like veterinarians or animal control. It's also possible that laboratory workers inhale aerosolized rabies, but again, this is very rare due to lab safety procedures. Other bodily fluids like blood, feces, and urine do not carry the virus. It's just the saliva of rabid animals. There are over 59,000 documented cases of rabies each year, and over 95% of these occur in Africa and Asia. 99% of all human cases are caused by dog bites, and most of these are domestic dogs. Rabies is found mostly in poor, disadvantaged populations with limited health care. This is due to the lack of rabies education, prophylaxis, and vaccination. Vaccination includes human vaccination, but also dog vaccination. According to the CDC, about 70% of dogs should be vaccinated in order to adequately eradicate domestic dogs as a reservoir. The highest number of human cases occurs annually in India, closely followed by many sub-Saharan African countries. The data represented on these maps is from 2017. The map on the top, labeled A, shows the total number of human deaths per year. According to this map, India has the highest rabies death rate, followed closely by China, the Democratic Republic of Congo, and Ethiopia. The map on the bottom, labeled B, takes population into account. It represents the number of human deaths per capita, which is per 100,000 people. India is still pretty high, but compared to sub-Saharan Africa, the percent of the population dying from rabies is a little bit smaller, and you can tell because it's not as bright red as some of the countries in Africa. In the United States, 92.4% of cases are reported in wild animals, and which wild animal depends on the geographic location. So in North Carolina, you can see on the map that most of the reported rabies cases are in raccoons, but overall for the United States, 30.9% of rabies cases are in bats, 29.4% are raccoons, 24.8% skunks, and 5.9% are in foxes. In the United States, there have only been 23 cases of human rabies in the past decade, and 8 of these cases have been contracted outside of the United States and its territories. Human rabies deaths have decreased significantly due to animal control programs and vaccination programs. Mass vaccination of domestic dogs has removed them as a reservoir, which has caused a decrease in dog bites in the United States, so therefore a decrease in human rabies. This graph shows the frequency of rabies cases over the years 1944 to 2002 in domestic animals, which is blue, and in wild animals, which is the gold, and also human cases, which is that orange line. The number of domestic animal cases looks to be directly correlated with human cases. As the domestic animal rabies cases decrease, so as the blue decreases, so does that orange line that represents human cases, which just makes sense because we are around domestic animals much more than wild animals. So if domestic animals have fewer domestic animals have rabies, then we're going to have fewer bites that allow humans to contract the virus. So let's discuss a little bit about the structure of the rabies virus. The single-stranded RNA genome within the rabies virus codes for 5 proteins. The first is a nucleoprotein around which the RNA is wound. The second is a phosphoprotein that's been shown to play a critical role in neuroinvasiveness, and it's been shown to work as a cofactor for RNA polymerase, binding it with the nucleoprotein that is directly bound to the RNA genome. The RNA-dependent RNA polymerase allows the negative sense RNA to be translated into mRNA so that the proteins for the progeny virions can be transcribed. The matrix protein makes up the viral capsule, and the glycoprotein is located in the viral envelope, helping with attachment to host cells. The glycoprotein, or G protein, is the only protein that can induce the body to produce neutralizing antibodies, which I will discuss later in this presentation. These 5 proteins make up 2 major structures. There's the inner part, the ribonucleoprotein core, also known as the RNP. This is made up of the RNA tightly encased by the nucleoproteins. It includes all the genetic information and all the proteins that aid in translation. Then there's the outer layer that's made from the matrix proteins surrounded by the envelope, where you have your glycoproteins forming trimeric spikes that aid with attachment to the host cell. In this image, you can see all of the 5 proteins we just discussed. The blue protein is the nucleoprotein, and the RNA is wrapped around, and it is the black line here. It's kind of hard to see. So the nucleoprotein binds the RNA, and then the phosphoprotein links the nucleoprotein to the polymerase as a cofactor, making it easier for the polymerase to bind its substrate, which is RNA. So here you've got your phosphoprotein, it's this orange little circle here, and then you have your polymerase here, which is your red. It just sort of allows the polymerase to be in the same vicinity as the RNA, so it makes it much more likely to undergo the reaction. You can see the gray matrix around the RNP core right here. And then on the very outside in the envelope, you have your trimeric spikes that are created by the glycoprotein, protein G. Typically, the virus begins its journey in the muscle tissue after a rabid animal bite. It then moves to the neuromuscular junction, entering the peripheral nervous system through a motor neuron axon terminal. It may also enter a sensory neuron if the bite is only skin deep and the virus isn't able to get to that muscle tissue. Rabies is known to bind to nicotinic acetylcholine receptors in the synapse, but has also been recently shown to bind to 2 other proteins called P75 neurotropin receptor and neural cell adhesion molecule. The virus then moves along the nerve axon back to the spinal cord. This is called retrograde axonal transport. The virus moves about 50 to 100 millimeters per day, so it will take varying times for symptoms to appear based on where the bite occurred. Rabies replicates within motor neurons of the spinal cord, dorsal root ganglia, and in the brain. Once the virus makes it to the brain, it then moves to the salivary glands and into the saliva, which is when an animal becomes contagious. So let's take a little bit of a look at the molecular mechanism by which rabies replicates. First, the glycoprotein on the outside of the virus binds to nicotinic acetylcholine receptors, which is what's happened here in number 1. And the virus then enters the cell through pinocytosis, which encloses the virus in a vesicle or an endosome. And right here, you've got this green organelle, that's your endosome. The low pH inside the endosome induces the viral envelope to fuse with the endosome membrane, which releases the RNP, which is that interior core that includes the RNA. So here you see that RNA genome is being released into the cytoplasm. Then viral polymerase translates the rabies negative sense RNA into positive sense mRNA, which can be transcribed by host enzymes to create more viral proteins, which is what you see happening right here. You're making more G, M, N, P, L, all these proteins. Or it can be translated back to the negative sense RNA as copies of the viral genome, which is what's happening right here. And that new copy of the genome will go into the capsule of a new progeny virion. So there are 5 stages of rabies. The first stage is the incubation stage, which averages 30 to 90 days, and it's completely asymptomatic. The second stage is the prodromal stage, which can last 2 to 10 days, where patients will have flu-like symptoms, as well as other symptoms such as vertigo, anxiety, irritability, increased libido, insomnia, hallucinations, and photophobia, which is being extra sensitive to light. The third stage is the acute neurologic period, which lasts about 5 to 14 days. It occurs when the virus has really taken a hold of the central nervous system and severely affects the functioning of neurons. Symptoms of this stage include hydrophobia, which is an irrational fear of water due to difficulty swallowing, paresthesia, and the abnormal dermal sensation. It's sort of like prickling or itching around where the bite occurred. Patients may have muscle spasms, seizures, hypersalivation, or paralysis. Rabies can manifest itself in 2 forms. It's known as either furious rabies or paralytic rabies. In furious rabies, infected individuals experience hydrophobia, and in paralytic rabies, on the other hand, this causes muscle weakness and paralysis. After the acute neurologic period, usually the patient goes into a coma and then dies due to respiratory arrest. Negri bodies are pathognomonic to rabies. They are masses of excess proteins clumped in the cytoplasm of nerve cells. They are typically found in the pyramidal cells of the hippocampus, the cerebral-Purkinje cells, and the brainstem nuclei, which is where the cranial nerves exit the brainstem. Originally, they were thought to be a byproduct of excess proteins produced during infection, but more recently it's been discovered that viral RNA along with viral polymerase are both present in these Negri bodies, which causes researchers to believe that they're actually little viral factories. In the image to the right, the darker circles are the nuclei, but the lighter purple mass within the cytoplasm is the Negri body. So there are vaccines for rabies. There are actually several of them, even though only 3 are administered in the United States. Rabies vaccines can be administered before or after a rabid animal bite. All bites from wild animals should be considered rabid, and a bite from a domestic animal, at least in more developed countries like the United States, usually does not warrant treatment, but the pet should be observed closely over the next 10 days to detect any symptoms of rabies. And if the pet does begin to show these symptoms, then the person bit by that pet needs to go into the doctor immediately. The rabies vaccine should be consistently given to those with occupational rabies risk or to those who live in or are visiting countries where rabies is widespread. The 3 vaccines administered in the United States are all equally effective and equally safe. They are given in 3 doses within 3 weeks, and you can get booster doses after a bite. The Rabivert is one of the vaccines. It's grown in a chick embryo cell. The rabies vaccine adsorbed, or RVA, is grown in a diploid cell line derived from fetal monkey lung tissue. And Emovax rabies vaccine is created using human diploid cells. All treatment for rabies is preventative because once symptoms appear, the disease is almost always fatal. After a rabid bite, cleaning the wound thoroughly with soap and an antiseptic called benzalkonium chloride can be very helpful. Rabies vaccines should be administered into the arm. If the person has not had a rabies vaccine before, then a 4-shot series is necessary if they've been bitten by a rabid animal. If they have had the vaccine before, then usually 1 shot is enough. Rabies immune globulin should also be given as close to the wound as possible. This provides neutralizing antibodies until the body can respond to the vaccine and then create its own. In the image to the right, you see how the rabies virus attaches to the receptor proteins on the cell surface, and it's taken in through endocytosis. Neutralizing antibodies are proteins that prevent the virus from entering the cell or from releasing its genome for transcription. In this new image, you can see how the immune globulin causes the aggregation of virions, or it can bind to the glycoproteins used for attachment. This blocks the virus from entering the cell. The immune globulin can also block the uncoating of the virion by preventing the fusion of the viral envelope with the endosome membrane. Those who are at constant risk of contracting rabies due to their occupation or location are encouraged to get rapid fluorescent focused inhibition tests, or RFFIT, consistently to monitor their rabies antibody levels. This will indicate when a booster dose of the rabies vaccine is necessary. The way the RFFIT test is performed is pretty interesting, and I think it's really creative. So first, different dilutions of blood serum of the patient are mixed with constant amounts of rabies. If the rabies virus neutralizing antibodies are adequately present within the blood, they will neutralize the viruses. The solution is then added to cultured cells and incubated. Any viruses left unneutralized will infect cells, and then this can be seen under a microscope with fluorescent dye. The more infected cells there are, the fewer antibodies there are in the blood. Doctors can use a reference serum to determine the antibody potency, which is expressed in international units per milliliter. The World Health Organization says that 0.5 international units per milliliter is an adequate response to the rabies vaccine, or complete neutralization at the serum dilution of 1 to 5. Here are some images of RFFIT slides. The one on the left shows many infected cells indicated by the binding of fluorescent dye. This proves that there were not enough neutralizing antibodies in the blood to keep the virus from infecting cells. On the right, though, you see no fluorescence, which indicates that there were enough antibodies in the blood to prevent the virus from infecting cells. The history of rabies in society is very rich and goes way back. Rabies is derived from the Sanskrit word rabhas, which means to do violence, which makes sense because rabid animals become violent and attack people. The earliest reference to rabies was around 2300 BC in the Mesopotamian creation myth. The goddess of chaos, Tiamat, was guarded by 11 monsters, one of which was a rabid dog. The Mesopotamian legal code also outlined a fine for failing to confine your mad dog if it bit and caused the death of another man. Democritus, an ancient Greek philosopher, wrote about the symptoms of a rabid dog, and Hippocrates, an ancient Greek physician, described symptoms of rabies in people, describing them as persons in a frenzy, drink very little, are disturbed and frightened, tremble at the least noise, and are seized with convulsions. Aristotle also referred to rabies in his writing Natural History of Animals. He said, "Dogs suffer from the madness. This causes them to become irritable and all animals they bite to become diseased." Louis Pasteur discovered the first rabies vaccine in 1885 when a 9-year-old boy was bitten by a rabid dog. He inoculated the boy 13 times with infected rabbit spinal cord, and the boy never developed rabies. Rabies shows up in more recent literature and film as well. Two examples are the well-known story To Kill a Mockingbird, written in 1960, where there's a scene in which Atticus Finch must shoot a rabid dog. Old Yeller, a film created in 1957, is all about a beloved dog who saves his owner from a rabid fox but is bitten in the process. There are many films, including several horror films, that include stories of rabies. There's a horror movie called Rabid that was made in 1977. I Drink Your Blood is a cult horror film from the 1970s. The film Rage from 1966. All of these depict very scary symptoms of rabies. Rabies has frightened humans for thousands of years and has just recently become less of a concern in the United States. The disease will continue to affect society until we can effectively educate and vaccinate the developing countries of the world. And here are my references. Thank you guys so much for listening, and I'm excited to see everybody else's presentation.